Yeah,
Yeah.
slide view. Let's see.
Okay, let's see. Slideshow.
Slideshow, yeah, yeah. And
And
then it says custom slideshow. Maybe that would do it. Let's try it. Yeah.
or from
Although,
the,
The first one I think.
yeah.
I don't know. So that pulled up the same.
Okay, something is up. Go back to what you just had. Yeah, you're okay. Maybe escape. Okay, good. You're good. Go ahead.
Well, so you let me minimize. You see the thumbnail of everybody who's sitting
Yeah, we're
in,
fine.
or
We're
just,
fine. Go ahead.
Okay, so I was gonna just talk a little bit about inflammatory bowel disease, and so I'll try to say click on here. And so that's what I've been investigating, and I'm currently switched over to the Division of Digestive Diseases here at UCLA, and sort of trying to start up sort of two separate axes of investigation, I guess I'll say. And I'll say a little bit about what those are and why I've chosen them. But first, I'll just give a little bit of background on inflammatory bowel disease. So inflammatory bowel disease consists of something called Crohn's disease and ulcerative colitis. And these are chronic and relapsing inflammatory disorders of the GI tract. Crohn's disease, which you can sort of see here on the left, it's characterized by inflammation that sort of works its way across or through it's sort of transmural. So it doesn't just include the epithelium, but it reaches sort of all the way even to the muscle. And it's often kind of localized here to the ileum, but it'll show up, it can show up in other places in the small intestine and other places in the colon. Like ulcerative colitis, it's a disorder of the colonic mucosa. It's kind of the upper layer of the layer that's closest to the lumen, the interior of the space of the intestine. And it always sort of starts here at the sort of rectal area, and it'll work its way back, you know, up the colon as it gets increasingly worse. And there are actually, you know, 1.6 million people in the United States that are currently affected by IBD. So if you look at the sort of increase over time in the United States, you know, it's almost got it's been sort of rising year over year. And it used to be that in Asia and Southeast Asia, there was very little inflammatory bowel disease, but with globalization, you see increasing prevalence even in Asia, and it's sort of got a bimodal appearance distribution. It will show up in younger people who are 15 to 25 for a first occurrence, but it also will show up in older individuals, 50 to 70 for the first occurrence. For the most part, ulcerative colitis is sort of weighted to this later appearance, and Crohn's disease is sort of will tend to show up earlier in patients. You know, the economic impact in the United States is it's sort of considerable. The cost ratio of respect to IBD has sort of been going up. You know, the cost has been going up over time. To some extent, that's largely due to the increasing use of biologics as a common therapy against IBD. But you know, a lot of money is spent on IBD annually. You know, just to look a little more closely at Crohn's disease here, again, like 30% of patients have disease sort of localized to the ileum here. Another 40% have involvement of the ileum together with portions of the, here's the ileum together with portions of the large intestine. About 20% have ulcerative colitis, have Crohn's disease, but it's sort of localized to the colon. The upper GI involvement, 5%. Perianal up to 30%. For ulcerative colitis, as I said, it sort of starts in this region. 30% of patients have ulcerative colitis limited here. Another 50% has involvement of the sigmoid colon, and then 20% have ulcerative colitis that reaches across the entire colon. A brief overview of the sort of clinical presentation of Crohn's disease. So, it can actually appear in any segment of the GI tract, and one of its clinical signs is actually an oral ulceration. Tends to spare the rectum. Can present as discontinuous inflammatory lesions. And it's a transmural inflammation that has a lot of sort of complications associated with it. So stricture or a severe narrowing of the intestine, fistula, perforation. You can get fairly severe abscesses. Here on the left is what normal tissue would look like, and on the right is Crohn's disease, and you see this sort of cobblestoning appearance of all this white, very inflamed sort of tissue. The pathologic changes. So you get inflammation that's not just limited to the epithelium and the mucosa, but it'll actually sort of reach all the way down into the intestine. It's got a cobblestone appearance, which you just saw in that last image. You can get granulomas that form. That's when you have a bunch of immune cells, often macrophages, which are sort of end up getting encased there. Here's a granuloma over here. You get chronic inflammatory infiltrate, and the crypts. That's the part of the epithelium or the lining of the single cell lining of the intestine, which is grows out of little vase-like structures where there are little stem cells at the base of that, and you can get sort of distortion, death of these crypts. You can get sort of crypts fusing together, crypt abscesses. So if you look at a biopsy sort of prepared histologically, you can see derangement of the epithelium. So, Crohn's disease, some additional clinical features, symptoms, diarrhea. You'll have colicky, abdominal pain, weight loss, fever, malaise, vomiting. Various signs there as well. And these are these complications that I talked about. You can get this thickening of the intestine here, which is sort of a function of the severe inflammation, and along with that, you can get stricturing, a kind of closing off of the in this case the ileum. You can have these sort of penetrating ulcers and form these strange, distorted ileal structures here. And what's sort of sad about Crohn's disease is that, even though there are sort of increasingly good biologic medications that are being used, up to 75% of patients will require surgery in their lifetime. The surgery just involves kind of just cutting out the inflamed intestine there. So even though the mortality associated with IBD is low, the morbidity or the kind of disease burden for patients is very, very high. And then just a couple words here about ulcerative colitis. It's confined to the colon. It begins in the rectum and extends proximally in a continuous fashion. It nearly always affects the rectum, and it can sort of work its way up the colon. And it's a continuous rather than skipping superficial mucosal ulceration. So it's confined to kind of the outer, you call it the inner layer of the intestine, the part that's immediately facing the lumen of the intestine. And here on the left is sort of normal colon, and here's highly ulcerated colon that you see in ulcerative colitis. You can get some of the same pathologic changes, crypt abscesses, crypt architectural distortion. Again, this is in the epithelial layer. And then and you can get, you get a lot of acute and chronic inflammatory cells in the region that's right under the epithelium. It's got a similar set of symptoms as Crohn's disease and sort of a similar set of signs. Complications, one of them is something called toxic megacolon here, which you can see on this image. That the colon gets huge and enlarged on an abdominal X-ray, and it's associated with fever, tachycardia, dehydration, and increase in white blood cell. You get perforation, bleeding. It's a significant risk factor for colon cancer. And so people that have ulcerative colitis are always getting surveilled for colon cancer. Just a brief slide here on Crohn's disease versus ulcerative colitis. Crohn's disease, again, it can affect anything between the mouth and the anus, whereas ulcerative colitis is the colon only. Crohn's disease is transmural, whereas ulcerative colitis only involves the mucosa or the epithelium and the little region below it. It can have lesions that are sort of skipped. You can have a lesion and then a healthy area and then another inflammatory lesion, whereas ulcerative colitis will have continuous inflammation. This has a bimodal age distribution, whereas ulcerative colitis largely affects older age groups, although it can present in younger patients. The one sort of difference upon examination is that rectal bleeding is less common in Crohn's disease, more common ulcerative colitis. What I said about granulomas, those are seen in biopsies of Crohn's disease, but they're rare for ulcerative colitis. And the treatments, the goal of medical treatment now is to achieve remission, and sort of not just the absence of symptoms, but also to try to achieve mucosal healing or a deep remission. And so currently IBD clinicians are trying to make use of these biologics, trying to make use of the older immunosuppressant medications to bring about mucosal healing. So here's kind of inflamed intestine, and here's healing. And and with healing come fewer hospitalizations, reduced need for surgery, reduced colon cancer risk, improved quality of life. Just a kind of an overview of FDA-approved medications for IBD. There were some sort of original set of medications sort of approved back in the 50s, 70s. And hydrocortisone is sort of a general immunosuppressant. But starting here in 1995 and then continuing on, there have been a sort of a new class of drugs that's in sort of common use now, and sort of more versions are in trials and coming online. But these are called biologics, and for the most part, they're antibodies that are directed at some aspect of the inflammatory process that is common across many or most people that are suffering from IBD. The very first sort of subclass of these that got approved and then developed over a series of iterations is something called anti-TNF biologic. Those are antibodies that are directed at tumor necrosis factor alpha, and that's one of the kind of most basic and most important pro-inflammatory stimuli that's sort of released early on within inflammation, but it helps sort of drive the inflammatory process. And so having these antibodies sort of target TNF and thereby inactivating it is proven to be helpful for a fair number of patients. You know, there are other biologics like this one here, anti-integrin based one. This actually targets the part of the immune cells that hook on to a particular receptor in endothelium that allows the immune cells to target the intestine. So with this particular medication, you're trying to prevent the influx of immune cells into the intestine. If you can sort of lower reduce the number of immune cells, you can thereby reduce inflammation as well. And then here it's just an early trial looking at anti-TNF agents, or several trials, but the ones that were achieving clinical remission, they had about 48% in this trial under this dose, 20 to 30% across several trials, and this was remission measured at 4 weeks. And if you combine anti-TNF therapy with an immunosuppressant, sort of AZA here, you can increase the degree of percent remission. But even under this combined therapy, you can see here that in these two trials, there were a lot of patients, 55% or approximately 40% of patients that didn't respond, didn't achieve remission. So there remains a lot of unmet need on the medication front. And just, a slide here on the limitations of anti-TNF alpha therapy is the most commonly prescribed biologic, but the monoclonal antibodies that get introduced are themselves immunogenic. So over time, the patient can develop a loss of response to these, and even prior to that, 20 to 30% of patients will be primary non-responders to therapy, and another 30% will experience a secondary loss of therapy, and so and no, I'll skip over that, but just to summarize sort of the different classes of biologics that are currently prescribed. So you have several that are trying to neutralize cytokines that promote inflammation. So those are, for example, the anti-TNF antibodies. You can try to neutralize cytokines. Those are these proteins that are these signals that get released by immune cells to sort of and other cell types to communicate with each other. You can try to neutralize cytokines that drive the differentiation and function of specialized immune subsets. In this case, they're antibodies against two particular subtypes of T cells, Th1 and Th17 specific T helper cells. You can try to block signal transduction cascades downstream of these inflammatory or differentiation pathways. So they're inhibitors of these signal signaling pathways, and then you can try to modulate lymphocyte or immune cell trafficking, and that's what I had mentioned, these anti-alpha 4 beta 7 integrin antibodies, and then and just for all these classes taken together, you know, up to 50% of patients develop resistance over time. And so combined with the high morbidity and the lack of success in achieving remission or kind of stable remission in up to 50% of patients across a lifetime, and again, there's a lot of unmet need. Let's see, I could talk a little bit about the mucosal immune system at homeostasis, but I'm not entirely sure for time, you know, how much time people want me to be speaking for. And so I might just say this about the GI immune system in general. You know, there are a lot of you know, a lot of bacteria in the immune system. I mean, in the colon, and as you work your way up through the small intestine, you know, you know, there are trillions of bacteria in the colon, and as you work your way up sort of towards the stomach, you know, the bacterial load decreases. But there's a, you know, huge surface area there in the intestine, and so you not only have these trillions of bacteria and bacterial products, but there's everything that we eat, there's environmental factors that we ingest, and so as a result, the GI immune system is sort of continually exposed to countless dietary and bacterial antigens and bacterial associated, like immune, you know, and inflammation triggering products, and so this kind of generates two challenges for the GI immune system. I mean, the GI immune system is sort of the largest immune system, I guess, in the body. It's the largest immune interface with the world in the body, and you know, and there are all those potential triggers of the immune system that are present within our intestine. So even though you know, there are all these bacterial components, most are harmless or beneficial, but the bacteria can quickly produce disease if they breach the barrier. And the barrier there is only a single cell layer thick, that's the epithelium. And so the first challenge of the intestinal defense and immune system consists of preserving the proper boundary between the commensal bacteria and the underlying immune cells. And, you know, but it's also, the digestive tract is the point of entry of various pathogens that can cause serious illness. And so you have all of these bacteria, which are, you know, we normally sort of are helpful or harmless, but they're, you know, they're mixed in, there can be pathogens. And so maintaining the proper balance there in the immune system between tolerance on the one hand, sort of tolerance against molecular patterns or antigens of commensal bacteria and food, while inducing an immune response against harmful microorganisms. You know, that's the sort of the central challenge of, or a central challenge of the GI immune system. And I won't say anything about this, but let me skip ahead through these slides. These are just sort of, if anyone's interested, I can go over this. I have a number of slides just on the GI immune system, but I'll kind of skip ahead to how inflammatory bowel disease develops. And in general, there's a schema that's sort of the received picture of IBD. So at the highest level of description, IBD is sort of thought to involve a dysregulated immune response to commensal bacteria. It can be brought about by environmental triggers, and it's to some extent in genetically susceptible individuals. So it's a little bit like an autoimmune disease, except that instead of the immune system sort of targeting self, as it would in a autoimmune disorder proper, it's responding or reacting to these commensal bacteria in a hyperactive way, and all of the the damage and the effects that we saw earlier there, the inflammation, the fistulae, the stricturing, the ulceration, that's all actually a consequence of our immune system. That's not because of pathogenic bacteria, but it's the overactivity of our immune system in responding to things that in most people it tolerates, that causes all of that damage. So there's something that's gone haywire in the immune system, and it's causing it to over respond to our normal gut bacteria. And and for these people, the damage is pretty severe. Again, like 70% of Crohn's disease patients are going to require surgery in their lifetime. With respect to the genetic component, to date, there actually been over 250. We call them genetic susceptibility loci, but these are alleles that seem to associate with a higher risk of developing IBD. The complication is what makes IBD very hard to study, I guess, and even to treat, is that first there are these over 250 risk alleles, but no single patient has more than a few of these, and so they're actually distributed across the patient population, but they're not present in every person, and what this seems to suggest is that there are lots and lots of different ways in which IBD can start. Downstream, once it's going, and once the inflammation becomes chronic, and once the damage starts to accumulate, the disease phenotypes across patients all seem to converge. They're very similar to each other. But the way the disease starts in patient 1 versus patient 2 can be very different. Patient 1 might have, PTGS2 here, that's COX2, might have a variation in like the COX2 promoter that leads to less transcription and translation of COX2. And patient number 2 might have a very different allele or susceptibility locus, and these, they, for these alleles, if you do analyses of the pathways they're associated with, they relate to a lot of the things that you would expect. They have to do with epithelial barrier and innate and adaptive immune system. But again, like they, between patients 1, 2, and 3, they might have very different risk alleles. And a further complication is that genetics actually doesn't account for all or even most of disease. So if it's the childhood cases that would most directly reflect a genetic component, but childhood onset IBD only represents 10 to 25% of all IBD cases. If you look at concordance rate in monozygotic twins, these are the ones that have exactly the same DNA. In Crohn's disease, 30 to 58% will develop Crohn's disease together, but in ulcerative colitis, only 10 to 15% of monozygotic twins will share. If one of the twins has ulcerative colitis, only in 10 to 15% of the cases will the other one have. And so it turns out that at most only 25% of the IBD disease can be explained by genetics. And so, and we saw this earlier on when I showed you the kind of increasing number of cases across time in both the West and over in Asia. Environmental factors play a very significant role. There's something associated with Western diet and Western lifestyle and with industrialization that that's contributing a great deal to the development of IBD. And there are many different environmental factors: pollution, smoking, infections, mode of birth, modern lifestyle, various socioeconomic factors, and so on, diet, and all those play important contributing roles. This is kind of a complicated slide, but I'll sort of talk through it for a few minutes, and it just sort of summarizes. This is from a New England Journal of Medicine article that came out, I think, in 2020, which is a pretty good review of IBD. If anybody's interested, maybe Mohammed can send out my email and I can send you the article. It's not that long, and it sort of gives a pretty good overview of inflammatory bowel disease. But in IBD, overall, we see a number of features that are associated with it. And the first is that you come to have a dysbiosis of the microbial, of our gut bacteria. Something about chronic inflammation sort of shifts the gut bacterial population. You get a reduction in the mucus layer. Normal mucus, like this is the small intestine, will be 2-layered, and you can sort of see it here. But in certain regions, you'll lose the mucus altogether. And that exposes this epithelial layer of cells here, sort of directly to the bacteria in the lumen and all these bacterial products. You end up getting increased microbial adherence to the epithelium, partly as a result. So normally the mucus sort of keeps the bacteria separated from the epithelium, but if you don't have mucus, you end up getting bacteria adhering to the epithelium. You'll get a dysfunction within specialized cells within this epithelium that play an antimicrobial role. So you get a loss of the products that they make that sort of tend to keep the bacteria at bay. You'll start to get a leakiness or a dysregulation of tight junctions and a loss of barrier function. And so with that leakiness, these bacterial products will kind of make their way into this region here called the lamina propria. And that's where all of the immune cells are. And that sort of results in increased antigen capture by dendritic cells because there's no mucus anymore, because bacteria are making their way into this region. The bacterial antigens or the protein pieces and peptides that end up getting presented to the adaptive immune system. There are many more of these things floating around. So the dendritic cells, whose job it is to sort of capture these antigens and present them in the lymph nodes, end up capturing more antigens. And then you end up getting macrophages that become activated, sort of pro-inflammatory macrophages. Normally in the intestine there, the macrophages sort of play an immunosuppressant and epithelial healing role, but they can also be very pro-inflammatory. And so the macrophages associated with IBD tend to be pro-inflammatory. And they produce a lot of pro-inflammatory cytokines, among them the TNF that I mentioned. The T cells in the normal T cell population in the gut there is sort of tolerogenic or immune suppressive. Those are called Treg cells or regulatory T cells. But in IBD, you end up seeing a lot of sort of pro-inflammatory T cells, Th1, helper T cells, also is Th17 subclass. And these sort of activated helper T cells, they're pro-inflammatory, and they recruit a lot of neutrophils and pro-inflammatory macrophages. And so you get a rush in of these neutrophils and macrophages, and so you end up with this highly inflamed region there that sort of starts with loss of mucus, the adherence of bacteria, the presentation of antigens, the loss of barrier function, and kind of activation of pro-inflammatory immune subsets. Let's see, that just kind of repeats what I said, but here's actually some work that I had done, and let me go to slideshow presentation just for a sec, even if it's a little bit harder to see, because the things will pop up in sequence here. But when I was studying how IBD arose in some mouse models that I was studying, I had observed that loss of epithelial barrier function ended up disrupting immune homeostasis and inducing severe intestinal inflammation in the presence of additional susceptibilities. In this case, COX-2, which I had mentioned earlier, was knocked out in the macrophage of mice. And if you just fed them a normal sort of, they call it a chow diet, the mice were fine. But if you gave them a diet that was high in fat and that also had extra bile acid in it, cholate, you ended up, so here's a COX-2 knockout mouse, eating the normal chow diet, and their kind of epithelium is healthy here, and these are these crypts, and this is, I'll just say that this is healthy looking. Wild type mouse, you give them this cholate diet, the cholate and high fat, and again, everything looks healthy. But you take the knockout mouse and you give it the cholate diet, and all of a sudden you get the loss of the epithelium, tremendous number of neutrophils and macrophages flooding in, and I had observed that actually this cholate diet induced an increase in barrier permeability that seemed to precede the development of inflammation. So loss of barrier function was a kind of initiating cause that was upstream of the development of severe inflammation in these mice. And in these mice, you ended up with a lot of epithelial damage, but that was actually downstream of the development of inflammation. So the first step was kind of an environmental factor that altered barrier function. In a normal mouse that had sort of normal genetics, that actually by itself didn't trigger an immune cascade, a kind of pro-inflammatory immune cascade, but if there was an underlying genetic susceptibility, in this case the loss of cyclooxygenase 2, which is the enzyme that's partially inhibited by aspirin or ibuprofen, or is inhibited by ibuprofen, then against that genetic background, these mice develop very severe inflammation. Kind of see here. So let me toggle back to that, and I had also looked at COX-2. This in the last slide, that was global COX-2 that was knocked out. It turned out that if you knocked out COX-2 in just macrophages, which are kind of innate immune cell, they're phagocytic cells, so their main role is to either ingest bacteria or ingest dying cells of our own body. But if you knock out, but they also will sort of trigger inflammation. They play a sentinel role in the intestine. Like if you knock out COX-2 just in macrophages and you give them that same high fat and cholate diet, that thing that that alters barrier function, you can develop the same very severe inflammation. And so, you know, here you can see that the muscle is very thin, and that here are these crypts I was talking about, and the little sort of villi or extensions from the crypts, and there don't seem to be a lot of immune cells. In contrast, this over here, all these cells that don't seem to be localized in any structure, those are all immune cells that have sort of rushed into this region. I'll just say something about the mechanism that I identified in this model. So here were the stain for like a bunch of neutrophils. I also had a stain for a bunch of macrophages, and so there seemed to be a lot of neutrophils and macrophages sort of together at the same time in these inflamed lesions. But normally, if you have acute inflammation, and that's what this little figure here represents, like, so let's say I don't know, you have a cut and bacteria get into the cut. The very first thing is you'll get swelling, and then you'll get an inrush of neutrophils, these kind of first responding cells of the innate immune system. And they don't live that long, a couple days, and then as they sort of start to undergo sort of programmed cell death, the number of macrophages kind of increases in the area, and these macrophages end up ingesting or eating the apoptotic or programmed cell dying neutrophils. And as the number of macrophages increases, the number of neutrophils kind of drops away altogether, and then the inflammation sort of resolves. But I wasn't seeing this in these mice. It was a mouse model. So that led me to think that maybe there was something wrong with resolution, inflammation resolution. And in particular, maybe there was something wrong with the process by which the macrophages were supposed to be clearing or ingesting, clearing these neutrophils. And then when I looked at some of the immune signals that were present there in the inflamed lesion, one of the things I observed was that this particular signal called lipoxin A4, which promotes resolution or inflammation resolution, which promotes this process by which macrophages ingest neutrophils, it was actually reduced in these mice. And then, you know, I did an experiment in which I added back an analog of lipoxin A4, and so here was the very inflamed mouse. But when I gave the inflamed mouse a kind of a version of lipoxin A4, it actually rescued or protected the mice from developing this type of inflammation here. And then in subsequent studies, I sort of observed that and this is going out in a current publication, that macrophages that are missing the cyclooxygenase 2, they're much less good at ingesting apoptotic neutrophils. So if you've got flocks, that's the kind of normal COX-2 mouse, you know, their level of neutrophil ingestion is kind of here. If you take away COX-2 from the macrophage, you lose a lot of that capacity to ingest apoptotic neutrophils. And along with that, or downstream of that, there was a loss of the ability of macrophages to bring about intestinal epithelial repair. One of the things that happens when these macrophages ingest neutrophils, it causes a kind of reprogramming in the macrophages, and they start to release a bunch of repair factors that signal to the epithelium and tell it to kind of repair itself, and that was missing in these mice. Or this is a kind of vitro experiment, but I won't explain the figure. It's a little complicated, but I'll just note that macrophages that didn't have COX-2, when they absorbed, when I challenged them with the dying neutrophils, they weren't actually able to repair the epithelium the way normal macrophages were. And here's just kind of a summary side on drugs, but I'll skip that. And then I'm just going to say two things about how I'm approaching investigating IBD going forward, and then I'll kind of wrap up. So I had mentioned that there are over 250 different genetic susceptibility loci that have been identified in human patients, and what followed from that is that no single patient, right, contains more than one or a few of these, and so there are lots and lots of different ways in which IBD can originate across a human population. Let's say you want to study IBD in mice. There's a lot of the research that we're doing happens in animal models. Well, most animal models are something you call monogenic, like they've got an alteration of a single gene. Like in my case, I took away cyclooxygenase 2 in, say, macrophages. And when I disrupted the barrier, the epithelial or the intestinal barrier in these mice, yeah, they developed a IBD-like inflammation. And I sort of worked on the mechanism there. But actually, there's only one very small subgroup of humans for which that mechanism is directly relevant, and it's a population in Northern Europe, sort of Scandinavia, where people have alterations in COX-2 promoter in this population, and they also developed IBD. So for that very limited population, this work in this single mouse model might be relevant. But you can't take 250 different mouse models and study them in a lab. It's way too expensive, way too time consuming. So you can't, if you're working off of a mouse model, like there's a way in which if you're trying to uncover the mechanism, you're working in a too narrow space. Like it's very, very hard to generalize across the human population because of that. And the other problem is just that the genetic, as I said, accounts for at most up to 40% of disease, and again, these mouse models, a lot of their disease is driven by the genetic component, and so it's hard to generalize from normal animal models, but the other issue has to do with translational or coming up with therapies. For it to be a viable or economically viable therapy, like it's got to apply to, a lot of, or most, or all of the human patient population. But if let's say I had this drug BML-111, which was a analog of lipoxygenase 4. Let's say that rescued disease in human patients that had a COX-2 related susceptibility. Would it apply to anybody else? Would it help anybody else? So how do you study the mechanism in a way that's general, that you can generalize over the human patient population, and how can you sort of identify therapeutic targets that would seem to hold for most people? And the biologics, that have been identified, the ones that people have been working on are general in that way. TNF, anti-TNF, why that was effective in about 50% of patients is because TNF is such a common pro-inflammatory cytokine. Why going after here modulation of lymphocyte tracking is that regardless of the first cause of IBD, at some point you get a huge influx of immune cells, and if you can sort of block that influx, that would help any patient, regardless of the first cause. So in my approach to IBD, I try to overcome the limitation of animal models. I've been sort of thinking at the level of secondary effects. I've been trying to identify effects that are common, are downstream of chronic inflammation, and thus might obtain for most or all people that have IBD, but secondary effects that that themselves seem to amplify or further drive disease. So can I identify a class of disease-driving secondary effects? You know, they're going to be, I'm going to overcome the problem of generalizability because they're going to be downstream effects of chronic inflammation, but they're going to be useful targets if, you know, if they're themselves further drivers or amplifiers of disease, they're going to be meaningful therapeutic targets. And I've got sort of two broad sort of avenues of investigation that satisfy that principle or that fall under that schema. And the first is I've observed, and others have observed, that there are a lot of oxidized lipids, free fatty acids that get generated non-enzymatically in these inflammatory lesions. And I've begun to demonstrate that you know, and they're downstream of having all these immune cells present with their sort of killing chemicals that get released to try to kill bacteria, but they also oxidize lipids. But these lipids can feed back into the innate immune system or the immune system, and among the things they can do is they can suppress macrophage efferocytosis, that process by which macrophages were trying to clear dying neutrophils. And so, you know, that's one class of things that I'm trying to target. And then I've also observed, and others have observed, that chronic inflammation can induce kind of secondary genetic changes. They're called epigenetic changes in intestinal epithelium. So exposure to chronic inflammation can result in, say, the adding of methyl groups. That's CH3 to various places in the DNA, and these actually can be passed down to progeny cells, and it can alter the gene families that get turned on or off in the epithelium, and I'm considering the hypothesis that these epigenetic changes sort of potentiate the epithelium to alter epithelial function in such a way that it promotes disease. So those are two types of secondary effects, but which I'm hypothesizing nonetheless help drive disease, and then maybe they're good candidates for general secondary disease driving mechanism, and they might, as a result, also be good therapeutic targets. So that's that was the. So I might stop there, and then from here on answer questions, or we all can talk amongst ourselves, or the like. And let's see, I'm going to try to pull up the screen in which I can see everybody. So that's what I have to say here. I can go back and speak more about the mucosal immune system more generally, or I can answer any other questions.